Hello everyone, hope you're all okay, you're all safe at home. So this week we're gonna study biology through a video, something new, huh? Okay, so the last time we started chapter 6, it was about the immune system. We spoke about how each one of us has our own HLA marker, the human leukocyte antigen. We spoke about the self and the non-self, and we spoke about the blood groups. Today's lesson is about cells of, or this week's lesson actually, is about the cells of the immune system, or what are known as leukocytes. Okay, so we all know the red blood cells, they're anucleated, which means they do not contain a nucleus. But the cells of the immune system are known as leukocytes, and they do contain a nucleus, so we call them nucleated. Okay, we could find them circulating in the blood or present in body organs. If they were present in body organs, we call them leukocytes, tissue leukocytes. If they were circulating in the blood, we call them circulating leukocytes. Okay, so we've got both in our bodies. Some are circulating and others are tissue leukocytes. Okay, they all originate from the same stem cell, a stem cell known as the pluripotent stem cell. This pluripotent stem cell could go into two different directions. It either goes to the lymphoid stem cell or the myeloid stem cell. If it was going to the lymphoid stem cell, the resultant would be a B lymphocyte or a T lymphocyte. So it differentiates, okay? It differentiates either into a B lymphocyte or a T lymphocyte. If it was a myeloid stem cell, it's gonna be one of three. Okay, it's either going to be a granulocyte, a monocyte, or a mast cell. Actually, granulocytes consist 67% of the leukocyte population, okay? And they are produced in large quantities and live 2 to 3 days only. I'm gonna go to document C for me so I could explain the different types of leukocyte. It's present on book page 122. Document C. So we've got three different types of granulocytes. We've got the neutrophil, eosinophil, and the basophil. You could see that we could differentiate them by the shape of their nucleus. This is the nucleus, okay? And neutrophil, I don't know. Anyways, it's up to you to take the shape. You could say it's horse shape if you want. Eosinophil, it's two-lobed, and basophil, it's really thinner than the rest. So each one of them has a different function. For the neutrophil, its role is phagocytosis, or eating up the microbes or the antibodies or whatever comes in, and destruction of bacteria. For eosinophil, we've got phagocytosis as well of antigen-antibodies complex. Okay, so an antigen is present. We have an antigen. Bacteria have different antigens. Viruses have another antigen. So whenever I've got a non-self antigen entering my body, my antibodies will go and neutralize it. They will clump against it. They will make a complex of antigen and antibody. Who will okay, it's not coming up. Who's going to phagocytose them? The eosinophil. The last one is the basophil, and it's responsible for the amplification of allergic reactions. So if you're allergic to something, this is the basophil are the reasons for the amplification of it. For example, the runny nose or the redness of the eyes during spring, this is an allergic response. What amplifies it is the basophil. This is if we're talking about the granulocytes. And these are the circulating leukocytes. If we take blood from anyone and make a blood smear, which is basically blood on a piece of slide, and we see it under the microscope, we're gonna see these three different cells. If we take a tissue, if we talk about tissue leukocytes, we're gonna talk about macrophages and mast cell. Macrophages are actually, if we go back to the myeloid stem cell, it's gonna differentiate into granulocyte, and it's one of the neutrophil, eosinophil, or basophil. Monocytes are later going to differentiate into macrophages. Okay, and mast cells are the same here. So we go back to the page. The macrophages make up, if I'm not mistaken, 6% of the leukocyte population in our body, and the role or the job of macrophages is again phagocytosis, or eating up the eating up, the gobbling up. The mast cell role is to release histamine. Later on, you will know that histamine has a major role in the first line of defense. We go to the lymphoid cells now. So we've got the B lymphocytes and the T lymphocytes. They make up 25% of the major leukocyte population. We distinguish them by their receptors. Receptors are like present on their membranes. In B lymphocyte, we've got antibodies, while on T lymphocyte, we've got TCR, or what's known as T cell receptor. Okay, so TCR stands for T cell receptor. Okay, TCR are receptors for antigens of other or for antigens of non-self, if you want. So whenever a B lymphocyte is active, so it becomes activated, it senses a pathogen, it senses something that's non-self, it differentiates into a plasma cell. And this plasma cell is what actually secretes the antibodies that go and attack or try to neutralize the pathogen or the non-self. I'm going to show you over here. So 1 is a B lymphocyte, okay? It's not active here. Once it's activated, it's going to change into 2, which is a plasma cell. This plasma cell is the one responsible for secreting the antibodies that fight the antigen of the non-self, okay? Letter C over here, or number 3 actually, not letter C, is a T lymphocyte. We've got 2 different types of T lymphocytes. We've got the helper T lymphocytes and the cytotoxic T lymphocytes. The helper T lymphocytes are known as TH, while the cytotoxic T lymphocytes are known as TC. Just like their name suggests, the helper T cell help other types of cells involved in immunity. They also express on their membrane a molecule known as CD4. You're not responsible so far for knowing what is CD4. You're only supposed to know that it's expressed on helper T cells. So far. Later on you will know what it is. But for now you're only supposed to know that T helper cells have CD4 on their membranes, and this is why we call them T4 lymphocytes, and they help other cells. Cytotoxic T lymphocytes, or TC, they actually kill body cells, not bacteria. They kill body cells that are infected by microorganisms. So let's say there's a body cell and then we've got bacteria coming in. This body cell is now infected with the bacteria or with the virus. So who kills it? The cytotoxic T lymphocyte. And the cytotoxic T lymphocyte express CD8 and not CD4. That's why we call them T8 lymphocytes. Okay, your homework would be to answer these questions. We're going to move on to document 5, and we're going to talk now about the lymphoid organs. Just give me a minute. What are the lymphoid organs and why are they important? The lymphoid organs are actually the site of production and maturation of lymphocytes. We've got 2 different or major lymphoid organs. We've got the primary ones and we've got the secondary. I didn't write it here. So we're gonna go through document A. Okay, so they made an experiment to know the role of the primary lymphoid organs. In this experiment, they had what's known as irradiation, which means destruction of dividing cells, or basically destruction of bone marrow. From now on, BM means bone marrow. Okay, so if irradiation occurred on a mouse, because in our case the experiment talks about mice, so it means that mouse in particular, or that lot of mice, has no bone marrow anymore. Ablation is the surgical removal of something, so they remove it surgically. So let's look at the experiment. We've got mice in lot A. What happened to them? Irradiation, so the bone marrow was destructed, and then graft of bone marrow. So they destroyed the bone marrow and then they grafted it. Okay, so they gave it another bone marrow. The result was production of B and T lymphocytes. In lot B, we had ablation of the thymus, plus irradiation, plus a graft of bone marrow. Immature T lymphocytes and B lymphocytes. So we have immature T lymphocytes and normal B lymphocytes. In lot C, we've got ablation of thymus plus irradiation. So I destroyed the bone marrow plus a graft of thymus, okay? So I removed the thymus, then I grafted another thymus. The result was there was no production of T or B lymphocytes whatsoever. Okay, so what can I analyze or what can I get out of this? The first thing, the primary lymphoid organs are the bone marrow and the thymus. The bone marrow and the yes. So, but what can you say? So, what's the job of the thymus and what's the job of the bone marrow? As we could see over here, an ablation of the thymus without it being grafted again resulted in immature T lymphocyte. So I could say that the thymus is the place or the site of the maturation of T lymphocytes. While over here in lot C, the ablation of thymus plus irradiation, so I removed the bone marrow or destroyed it, and then I grafted the thymus. I had no production of T or B lymphocytes whatsoever. So this tells me that the bone marrow is responsible for the production of T and B lymphocyte, and it is the maturation site of B lymphocyte, because over here in lot B I had only immature T lymphocyte, but the B lymphocytes were mature. Okay, so the first two, the primary lymphoid organs are the thymus and the bone marrow. The thymus is responsible for the maturation of T lymphocytes, hence T. The bone marrow is responsible, first of all, for the production of both T and B lymphocytes, and later on for the maturation of B lymphocytes. I want you to go to your book right now. Lymphocytes maturation. How do they mature? So if they do not mature, they are not functional. And this is what's known as immunocompetent. So they have to mature to become immunocompetent. So what happens during maturation, or how do lymphocytes mature? So before any encounter with non-self antigen, there are specific membrane receptors present on each lymphocyte before maturation. But what happens later on, so I've got several lymphocytes, each having a different specificity, okay? So they all carry membrane receptors. They now undergo screening. In the bone marrow, the B lymphocytes, whose receptors can bind self-antigens, are eliminated. So whenever I've got a B lymphocyte which can bind to a self-antigen, it is directly eliminated or killed or whatever you wish, but it's definitely not present anymore, so it does not mature. Why do you think this is important? Remember, B lymphocytes produce antibodies. As a result, if the antibody B lymphocyte can bind to self-antigen, it will be attacking the self. Okay, this is why all B lymphocytes who have receptors that can bind to self-antigens are eliminated. Okay, what happens now in the thymus? We have a double selection is occurring in the thymus. So the T lymphocytes, baby T lymphocyte, travel to the thymus where they're going to mature. The first selection occurs when all receptors that can bind to HLA molecules of the self are preserved. You will know why later. I need to keep the T lymphocytes that can bind to the HLA molecules of the self. And this is important because later on you will see how T lymphocyte fights infections or fight diseases or fight non-self in general. Okay, while others are eliminated. So all T lymphocytes who cannot bind to HLA molecules of the self are eliminated. All those who can bind to the self, HLA molecules of the self, are preserved. The second selection occurs when T lymphocytes whose receptors can recognize self-peptides are eliminated, while others are preserved. So the T lymphocytes whose receptors can bind to HLA molecules of the self are kept, but those who bind to self-peptides are eliminated. Again, you will know why later on. You could try to guess now. This is part of the mechanism that the T lymphocyte carries in order to fight non-self, which we will discuss later on. Okay, we go now to the secondary lymphoid organs. And the secondary lymphoid organs, we've got the lymph node, the lymph nodes, which are spread all over the body, as you could see. You see these tiny circles over here, over here. Yeah, these are all lymphoid nodes. And the spleen. And you've got the lymphatic network. So we've got the blood vessels for blood and the lymphatic network for lymph. What's lymph exactly? Lymph is extracellular fluid that is Okay, so lymph again is extracellular fluid that is extracted from the tissues. Okay, so let's say from the liver, from the heart, there's fluid coming out, so it's extracted. This fluid is known as lymph. So this lymph travels through the lymphatic network, passing through the spleen and passing through, not the spleen, sorry. It passes through the lymph nodes. The spleen has another role now. We will talk about it in a minute. So the lymph nodes are small organs lining up like a string of beads along a network of vessels called lymphatics. The lymphatics network collects extracellular fluid from the tissues and brings it back to the blood vessels. Okay, so what's the job? So why do I need to collect lymph from the tissue? Because if there is any non-self, okay, it will appear in this lymph. It will be extracted from the tissue and it will travel to the lymph node where all the action occurs. This is the sites of responses against antigens drained by the lymph. Okay, so the action occurs in the lymph node. The sites of responses against antigens drained by the lymph. So let's go now to the spleen, which is located in the upper left abdomen. Okay, the spleen is the site of reactions against antigens carried in the bloodstream. So I've got the lymph nodes for antigens carried in the lymph and the spleen for antigens carried in the bloodstream. Lymphocytes circulate continuously through the lymph and the bloodstream between the different secondary organs in order to maintain a permanent surveillance of the entire body against foreign elements. This is the biology lesson for this week. Hope you understand it. Again, if you have any question, you've got my email. Your homework would be probing the documents, book page 122, 1 through 4, and book page 124, 1, 3, 4, 5, and 6. So we only exclude 2, which you have to draw a diagram illustrating the maturation of B and T lymphocytes. Goodbye. Stay safe.